Assalamu alaikum students. A very good morning. I hope everyone will be fine. Congrats once again. As many of you have got much better grades now as compared to the previous results. But once again, forget about these grades now for A2 because your A-level result is going to be based on your A2 grades only. And this year, it seems like that the papers might be more tough, so the threshold will be very high because they are going to give you the grade just on the basis of 2 papers rather than 5 papers, right? So forget about these grades now. Whatever you have got, just concentrate on your studies in A2. So in the previous lesson yesterday, we talked about the photosystems, and then we talked about then we started with the stages of photosynthesis. And the first stage we talked about, that was the light dependent reactions. In that we talked about the cyclic photophosphorylation, then non-cyclic photophosphorylation, then photolysis of water. And remember that in the photolysis of water, it was there that the photolysis of water can be demonstrated or it can be investigated by the Hill reaction. Okay? So today we are going to talk about what is the Hill reaction and what happens in the light independent reactions of photosynthesis. So coming to the Hill reaction first. Actually, why this reaction is called as the Hill reaction, first of all, coming to this, there was a scientist, Robert Hill. Actually, he worked out this, reaction, and that's why this reaction is named after his name, the Hill reaction. Okay. So before coming to the Hill reaction, generally talking about the redox reactions. Obviously, redox reactions you have read in chemistry very well. We know the oxidation reduction reactions. And what is oxidation? That is the loss of electrons. And what is reduction? That is the gain of electrons, right? And when we talk about the redox reaction, we say these are the reactions which involve the transfer of electrons from a reducing agent to an oxidizing agent. Okay. So sometimes when we talk about the removal of hydrogen atoms, obviously the removal of hydrogen, that is called as dehydrogenation. Okay, so sometimes hydrogen atoms are transferred, right, between the two agents. So then we say dehydrogenation, that is equivalent to oxidation. Okay, so dehydrogenation, which is the removal of hydrogen, that is equivalent to oxidation. So basically, Robert Hill in 1939, he showed that the chloroplast which have been isolated, obviously, we have to isolate the chloroplast from the leaves, right, from the leaf cells. So he showed that, he demonstrated that once the chloroplasts, they are isolated, they had the reducing power. Okay, so reducing power was shown by using a redox agent, right? And what happened that when this redox agent was used, it changed color on reduction. When it became reduced, it changed the color. Now, this technique, basically the Hill reaction, it can be used to investigate the effect of two factors on the rate of photosynthesis. It can be either the effect of light intensity or light wavelength, right? So basically, because basically reduction यहाँ पे show कर रहा है, because actually there is a breakdown of water molecule. Okay, and as a result of that, hydrogen ions are released, which are going to be picked up by a hydrogen carrier molecule. This is what we are talking about in the natural conditions. But now we say, rather than NADP, we are using some redox agent, okay, to investigate this reaction once the chloroplasts, they have been isolated from the plant cells. So this technique can be used, so that's why it can be used to investigate the effect of two factors on the rate of photosynthesis. Either the effect of light intensity, because why we are talking about the light intensity or the light wavelength, why not other factors? Because basically for the photolysis of water, what is required? Light energy. Photolysis of water cannot take place in the absence of light energy. So we can investigate the effect of these two factors by using this technique. First is the effect of light intensity, second is the effect of light wavelength on the rate of photosynthesis. Okay, and what we can use for that, obviously we can use a suspension of chloroplast for this. Now basically Robert Hilt at that time, he used uh, these ferrikines as an acceptor. Okay, but rather than using these ions now, we can use various other redox agent, and a very good redox agent to be used for this purpose is DCPIP. We talked about DCPIP earlier as well. DCPIP stands for dichlorophenol indophenol, right? And you know that when DCPIP is in oxidized state, it is dark blue in color, but once it becomes reduced, it becomes colorless. So DCPIP can substitute NADP. Okay, because NADP is there in the natural conditions, but once we have isolated the chloroplast now, okay, so obviously we have to use some other technique which can be observed, right? And for that we are going to use some redox agent such as DCPIP, which can substitute the NADP. And DCPIP, once again, it becomes colorless when reduced. This is the equation for this. You can see over here, this is the oxidized DCPIP, which is blue in color. And when it is DCPIP, when used with the, like, obviously the chloroplast suspension that is mixed with the solution of DCPIP, and once the process has occurred, and after that DCPIP, when it becomes reduced, it becomes colorless. Okay, and you can see over here what is taking place over here, that is the photolysis of water. Now investigating the Hill reaction. Now what we can do for this, how it can be performed. First of all, we have to isolate the chloroplast from a leafy plant. Now the leafy plant, obviously we have to take those leaves which are showing very the green in color. So for that we can use the spinach leaves, we can use the lettuce leaves even, right? So what we are going to do, we are going to take uh, some of the spinach leaves, and then we are going to crush those spinach leaves using a mortar and pestle, right? And but we have to use the and but then we are going to liquidize this extract. We can take out the extract and then we can liquidize the extract. For making an extract, what we can do, that to the spinach leaves we can add some ice cold buffer solution. Okay, so we are going to pour the extract, and we are further going to liquidize it if required with ice cold buffer, and then we are going to filter that suspension, or we can centrifuge that suspension even. Obviously filtration is going to, with the help of filtration we are going to take out the filtrate, but once we centrifuge the suspension, actually centrifuging the suspension, it is going to remove the unwanted debris. Okay, so either way can be used, either we can filter or we can centrifuge that chloroplast suspension. And remember that we have to we have to keep two things in mind while investigating the Hill reaction. First, we have to perform this investigation as quickly as possible. Secondly, we have to use, wherever we will be, wherever you will read out, you will see, we have earlier used this ice cold buffer. So it's a buffer solution kept at a very, very low temperature, a chilled buffer solution. Then we are writing here again using chilled glassware. Whatever we are going to utilize for performing this investigation, whether they are the small tubes, whether there's a capillary tube, whatever is going to be utilized, it has to be a chilled glassware. So basically, why a chilled glassware, and why performing this investigation as quickly as possible? That is just to preserve the enzyme activity. Okay, So once we have centrifuged the suspension, or once we have filtered the suspension, what we are going to do, we are going to take the small tubes, and in those small tubes, obviously we have to add the buffered solution. Why to add the buffer solution? Because that is going to maintain the pH, right, which is very, very important to be kept constant during this investigation. So we are going to take this, because already to this suspension, you know, we have liquidized the leaves in ice cold buffer. So we have already added the buffer solution to that suspension, right? So what we are going to do, we are going to take those buffered class chloroplast suspension in the small tubes, and then we are going to add DCPIP solution in those small tubes. So what those small tubes contain now, these small tubes, they contain buffered chloroplast suspension plus added DCPIP solution. Then we are going to place these small tubes in different light intensities if we are investigating the effect of light intensity on the rate of photosynthesis, or we are going to place these small tubes in different wavelengths of light if we are investigating the effect of wavelength of light on the rate of photosynthesis. But remember that if we are investigating the effect of light intensity, then we have to keep the wavelength of light constant, and if we are investigating the different effects of different wavelengths of light on the rate of photosynthesis, then we have to keep the light intensity constant. Okay, so you see, once these small tubes are placed in different light intensities, are placed in different wavelengths of light, then we will see the blue color of DCPIP. It is observed at intervals, and we will see that the DCPIP color is going to be decolorized in some interval of time. So rate of loss of blue color is a measure of effect of factor being investigated, either the light intensity or wavelength of light on the activity of chloroplast. Okay, this is, you can see how we can show it graphically. You know, this is a, you know, what we can do, that as I said earlier, that the rate of loss of blue color, that is that is a measure of the effect of factor being investigated, either light intensity or the wavelength of light on the chloroplast activity. But remember that that rate of loss of blue color, it is either measured in a calorimeter, either it can be measured using a calorimeter, or we can measure it by matching those tubes against some known concentration of DCPIP solution. Okay, so this is how we can calculate the rate of loss of blue color, right? So this is, they have shown it with the help of a calorimeter, when the readings are taken with the help of the calorimeter, and you can see the calorimeter reading is proportional to the amount of DCPIP which remains unreduced. Now you can see over here there are two scenarios. One is the scenario when the chloroplasts are placed in light, and one is the scenario when the chloroplasts are placed in dark for about 5 minutes and then they are placed in the light. So you can see over here, when the chloroplasts are placed in light, you can see this is the blue color and this is going to be the colorless, right? So this is the calorimeter reading. Now you can see over here, after about 5 minutes, in 5 minutes, this is DCPIP is going to be decolorized. But when chloroplasts are placed in dark for about 5 minutes and then placed in light, you can see DCPIP is going to be reduced, but it's going to take more time here. Okay, roundabout, you can see the time over here as well. So this was all about the Hill reaction. I hope it's clear. Usually I never see any question related to this graph, not at all, particularly this graph. Okay, sometimes the Hill reaction can be there in paper 5, it can be there, sometimes in paper 4 can be there, right? But very, very rarely. So now we are going to talk about the light independent reaction of photosynthesis. Remember that light independent reaction, actually it involves only one cycle, and that is called as Kelvin cycle. Okay, now this cycle, why this is called as the Kelvin cycle, actually there were three people who worked out this cycle. They were Kelvin, Benson and Basham, and it was between 1946 and 1940 1953, but the major contribution was given by Kelvin in this case. So that's why this cycle is usually called as Kelvin cycle. Now, what happens in the Kelvin cycle? Actually, the fixation of carbon dioxide takes place in Kelvin cycle, which is a light independent process. Now, what happens in this? That I will move to this cycle now. This is a diagrammatic representation of Kelvin cycle. Now you can see over here that a carbon dioxide, now carbon dioxide is having one carbon. Okay, remember this story of, you know, number of carbons once again. So carbon dioxide having one carbon, it combines with a 5-carbon compound, which is ribulose bisphosphate. Ribulose bisphosphate, which is usually abbreviated as RUBP. Now remember one thing, whenever you are going to use the abbreviation, first of all, if it is a one-word answer, okay, like let's say they say name 5-carbon compound in the Kelvin cycle with which carbon dioxide combine. Now you have to write down ribulose bisphosphate. Never write it as RUBP. You are not going to get the mark. There are some rules and regulations of using abbreviations in Cambridge papers. Okay, and what are those rules and regulation? First of all, whenever there is a one-word answer, or that word is not going to be repeated. Let's suppose it is a single sentence answer and you are not going to repeat the word again in that sentence. It's going to be used once. So in that case, you are going to write down the full form of that, like as ribulose bisphosphate. However, if there is an essay question, or maybe there is a paragraph of 5 to 6 lines and you have to use this word of ribulose bisphosphate again and again, once in the very start, when you are going to use this word for the first time, you are going to write down the full form. Then in the bracket, you can write down the abbreviation. And then in the paragraph, when you are going to write about this compound again and again, then you can use the abbreviation. Okay, so I'm sure it's clear now. So carbon dioxide, which is one carbon, it combines with this 5-carbon compound, which is ribulose bisphosphate. As a result of that, a 6-carbon compound is formed, but this 6-carbon compound is highly unstable. So as soon as this unstable intermediate, which is a 6-carbon compound, it is formed, it's going to break up immediately into 2 molecules of 3-carbon compound. Now what is the name of that 3-carbon compound? That is glycerate 3-phosphate. Okay, glycerate 3-phosphate. Remember that this glycerate 3-phosphate, it is also sometimes called as phosphoglyceric acid. Okay, so glycerate 3-phosphate is GP, but sometimes it is also called as phosphoglyceric acid, abbreviated as PGA. Now, as these 2 molecules of glycerate 3-phosphate are formed, then this glycerate 3-phosphate in the presence of ATP, like obviously ATP is going to be hydrolyzed, ATP is going to be hydrolyzed into ADP and inorganic phosphate. So obviously using ATP and reduced NADP, and now you see, now you remember that both reduced NADP and ATP, they are the products of light dependent reaction. Okay, and they are going to be utilized over here in this way, right? So glycerate 3-phosphate in the presence of ATP and reduced NADP, it is reduced to triose phosphate. Obviously there are 2 molecules of glycerate 3-phosphate, so they are reduced to 2 molecules of triose phosphate. Once again, it's a 3-carbon compound abbreviated as TP. Okay, now you see this, what happens to this TP now? Most of the triose phosphate, and that is about 5/6. You can see this is the major quantity of triose phosphate. Okay, so most of the triose phosphate, which is about 5,6, that is used, you can see over here, that is used to regenerate ribulose bisphosphate. And for this process, once again, energy is required, and that comes from the hydrolysis of ADP into ADP and inorganic phosphate. Okay, so about 5/6 of triose phosphate that is used to regenerate ribulose bisphosphate. What is left behind? That is the 1/6 of triose phosphate. So what happens to this 1/6 of triose phosphate? Some of this triose phosphate, it condenses. Okay, and when it condenses, it forms the hexose phosphate. Okay, and hexose phosphate, obviously it is 6 carbon, which is in turn used to produce starch for storage purpose. It is used to it is used for the formation of some of it becomes the sucrose, and sucrose, it is going to be transported for the translocation purpose in the plant, around the plant body, or cellulose for making the cell wall. And obviously glucose is also there. It is one hexose phosphate, so it is used to produce the glucose is there, which is used in the process of respiration, right? And then rest of that triose phosphate, that is used to that is going to be converted to the glycerol and fatty acid, and the glycerol and the fatty acids, they combine together to form the lipids, and they can be used for the cellular membranes, or they can be converted to the acetyl coenzyme A, and you know that acetyl coenzyme A is used in the process of respiration, or maybe they are used for the production of amino acid for protein synthesis. So you can see how important is the triose phosphate. It's not only because when we are talking about photosynthesis, we are always talking about glucose formation, but you see over here, glucose is actually the main, because the main compound formed there. Okay, but otherwise you can see how triose phosphate is used for the formation of so many other compounds. Okay, so this was all about the Kelvin cycle. Now one very important thing, this process, this step of Kelvin cycle, where carbon dioxide, that combines with ribulose bisphosphate. This combination of carbon dioxide with ribulose bisphosphate, this is catalyzed by an enzyme which is called as ribulose bisphosphate carboxylase. This is abbreviated as Ribisco. Remember that Ribisco is the most common enzyme in the world. Obviously plants सबके बहुत ज़्यादा हैं दुनिया में, ठीक है, as compared to the other living organism. So that's why Ribisco is the most common enzyme in the world as well. Okay, now we will just have a look at the outline of this Kelvin cycle, starting from this, that carbon dioxide combines with a 5-carbon sugar, ribulose bisphosphate. A 6-carbon compound is formed, but this 6-carbon compound is highly unstable, so it immediately breaks up to give 2 molecules of a 3-carbon compound called glycerate 3-phosphate. Enzyme ribulose bisphosphate carboxylate, which is abbreviated as Ribisco, it catalyzes the combination of carbon dioxide and ribulose bisphosphate, and this is the most common enzyme in the world. Glycerate 3-phosphate, which is formed in the presence of ATP and reduced in ADP, it is reduced to triose phosphate. Most of the triose phosphate, about 5/6, is used to regenerate ribulose bisphosphate. The remaining, which is 1/6, that is used to produce other molecules required by the plant. Some of these triose phosphate condense to produce hexose phosphate. Hexose phosphate used to produce starch for storage, sucrose for translocation, cellulose for making the cell wall. Then other triose phosphates, they're converted to the glycerol and fatty acids to produce lipids, or acetyl coenzyme A for use in respiration, or in the production of amino acid for protein synthesis. So this was all about the Kelvin cycle. In our next lesson, we will be talking about the structure and the function of chloroplast. Then the factors, various factors which are going to affect the process of photosynthesis, right? So till then, Allah Hafiz, take care, have a good day.